I may just give an exception over here. The exception is insulin-like growth factors. Insulin-like growth factors are the mediators of growth hormone action. These are proteins themselves, but they are bound to proteins in the plasma. So this is an exception to this rule, and therefore, in fact, there are 6 types of insulin-like IGF-BPs, insulin-like growth factor binding proteins, IGF-BPs in the plasma. There are 6 types of these binding proteins, and therefore insulin-like growth factors have longer half-life. So you must remember always that the growth hormone has got short half-life in the plasma, only about 1 to 2 minutes, but in that short half-life, it will reach the liver and it will cause secretion of these IGFs, insulin-like growth factors, and the result would be then they will come into the secretion and they will have a long half-life to exert or mediate the action of growth hormone. So that's an exception. Remember, growth hormone, short half-life as it's a peptide hormone, but growth hormone mediators have longer half-lives, but with an exception to be kept in mind. So that's one more comparative feature. Next, well, let me just also talk about the synthesis of the peptide hormones on the sidelines of this discussion. As we have said that steroid hormones are synthesized from cholesterol, and the common step is cholesterol forms pregnenolone and pregnenolone forms progesterone, and from that step various other steroids are synthesized. Coming to the peptide or protein hormones, they are synthesized in the precursor forms. Synthesized in the precursor forms. I'm talking particularly about the peptide or protein family of hormones. So, precursor forms in which they are synthesized, and then they will be cleaved to form the final hormone, which will then come into the circulation. Let me give you some varieties in this. One variety is that a single copy of the hormone will be finally secreted from the will be finally released from the precursor hormone. So for example, insulin, single copy of the hormone released. Insulin is synthesized as pre-pro-insulin, then it will be cleaved to form proinsulin, and then proinsulin will finally form one copy of insulin and C-peptide, the connecting peptide or C-peptide, and then that will be then released. So single copy of the hormone released from the precursor. Second, multiple copies of the same hormone can also be formed from the precursor. How so? The example is TRH. TRH is a tripeptide. It is synthesized from pre-pro-TRH, thyrotropin releasing hormone, and once it's cleaved, the multiple copies of the TRH, 6 copies are formed from a single precursor of TRH. So multiple copies of the same hormone. And the third variation in this is that variety of hormones derived from the single precursor. So multiple copies of the same hormone, the example was TRH, and multiple hormones formed from a single precursor, that's also possible. Example here is POMC, pro-opiomelanocortin. Pro-opiomelanocortin. Pituitary basophils synthesize this large polypeptide precursor. Its first proteolytic cleavage will form corticotropin or ACTH and beta LPH, lipotropic hormone. Further cleavage will form MSH, CLIP, that is corticotropin like intermediate lobe peptide, and on this end the cleavage will form endorphins. So multiple hormones formed from a single precursor, that also is possible with these peptide or protein hormones. So just an additional feature about the synthesis of the peptide or protein hormones. Coming to the further comparison, as we were comparing the peptides versus steroid hormones, peptide proteins or some hormones belong to the glycoprotein structure family. Next point of comparison is that once a final hormone is formed in the peptide or protein category, it is generally not modified further peripherally. It's the final hormone and it's going to act on the target cells. In the case of steroid hormones, even if the hormone is formed finally, when it goes to peripheral structures or target tissues, it can get further modified. Classic example of this is let's say testosterone. Testosterone is converted into its metabolite DHT, 5-DHT. So even that is active. So testosterone is active and its metabolite is also active. So peripheral conversion may form an active metabolite. That's possible with steroid hormones but not with the peptide hormones. Another comparative feature. Next is let's say mechanism of hormone action. Now let's talk about the mechanism of action of these hormones. We know that these hormones, they are going to act via their receptors. Now since peptide or protein hormones, they are not lipid soluble, so they won't be able to enter the target cells easily, because as I have already mentioned, the cell membranes are made up of lipid bilayer and these are not lipid soluble, so they won't be able to enter the target cells easily, and therefore their receptors are located in the cell membrane itself, so that the hormone does not have to enter the target cell. Receptors in the cell membrane. Whereas steroid hormones, their receptors are intracellular receptors. Let me make this point clear. The steroid hormones have intracellular receptors, which means because steroids are lipid soluble, they can enter the target cells easily, and therefore the receptors are inside the cell. Now the receptors can be inside the cytoplasm or nucleus. So cytoplasmic receptor or nuclear receptors. For instance, estrogen is known to have a nuclear receptor, but then recent evidence indicates that even though estrogen or other steroid hormones, they have intracytoplasmic receptor, what really happens is that this is a primary receptor. So the steroid hormones enter the cells. Here is a steroid hormone. It will enter the cell easily crossing the lipid bilayer. It will combine with the receptor, and then this hormone receptor complex will enter the nucleus. So anyways, the steroid hormone is going to enter the nucleus, whether combined with the primary receptor. For estrogen also, as I mentioned, there is an intranuclear receptor, but there have been primary receptors which have been found in the cytoplasm as well. Recent evidence indicates that. However, the point is that it's an intracellular receptor, means receptor inside the cell, whether it's cytoplasm or in the nucleus. And then when steroid hormone combines with the receptor in the cytoplasm, then that hormone receptor complex, let's say this is a hormone receptor complex, will enter the nucleus and will attach to the specific sites on the DNA strands. They've been described as hormone response elements, HREs, or hormone response elements, and eventually the action of that hormone will be elicited. So that's about the steroid hormones. Basically, intracellular receptor, whether cytoplasmic or nuclear receptor. Well, let me just add here that thyroid hormones have intranuclear receptors. The third category of hormones that we discussed, peptide hormones, steroid hormones, and derivatives of a single amino acid. And in that third category, it was thyroid hormone and catecholamines. So let me just complete this by saying thyroid hormone has intranuclear receptor, and catecholamines, their receptor is in the cell membrane receptor. So let me come to this phenomenon of cell membrane receptors. The cell membrane receptors are of two types. Basically, two types of cell membrane receptors. One is a G protein coupled receptor, and the second type is catalytic receptor. This is specifically for the cell membrane receptors. This is specifically for the peptide or protein hormone receptors. Two types of cell membrane receptors for them: G protein coupled receptor and catalytic receptor. So far as the G protein coupled receptor is concerned, you know how it works is that when the hormone comes to the target cell, there is a receptor which is associated with the G protein. Now, therefore, this is called as G protein coupled receptor. What about this G protein? G protein, also called as GTP binding protein, it has three subunits: alpha, beta, and gamma. Now, it's also called as heterotrimeric G protein because the structures are different for these subunits. So heterotrimeric G protein with alpha, beta, gamma subunits. So when hormone combines with the receptor in the cell membrane, now alpha subunit of the G protein dissociates from the beta gamma subunits, and it moves along the membrane, this alpha subunit, and then it will activate an effector protein inside the cell. For instance, adenylyl cyclase enzyme will be activated. This will then convert cellular ATP into cyclic AMP, and then the cyclic AMP, which is called as a second messenger, it will activate the various protein kinases. Those enzymes will act on various amino acid residues on intracellular proteins, and that is how the hormone effect will be exerted. So cyclic AMP is the second messenger for all these hormones like ACTH, TSH, FSH, LH, PTH, glucagon. All these peptide hormones or protein hormones, they have cyclic AMP as the second messenger. Well, if guanylyl cyclase enzyme is activated, then cellular GTP will be converted into cyclic GMP. Cyclic GMP can be the second messenger for a few hormones, particularly ANP, atrial natriuretic peptide, and NO. These hormones are known to have cyclic GMP as a second messenger. So that's a G protein coupled receptor and its mechanism of action. The other, like catalytic receptor. Now in this category, we have two important types of receptors. One is receptor tyrosine kinase, also called as RTK, receptors tyrosine kinase or tyrosine kinase receptor. Example, insulin. Insulin has tyrosine kinase receptor. Now insulin receptor has alpha beta subunits, two subunits, alpha and beta, and when insulin binds with its receptor, with the alpha subunit of the receptor, then there is autophosphorylation of the receptor, and then there will be activation of tyrosine kinase. So it will act on the proteins inside the cells. That's a tyrosine kinase receptor. Whereas growth hormone receptor is slightly different than this. It is described as tyrosine kinase associated receptor. Please try to understand that I am putting both these receptors in the catalytic receptor category because the receptor itself has catalytic activity, or it by itself activate the enzyme. Whereas in the case of G protein coupled receptor, receptor did not move. It was the G protein and its alpha subunit which moved and which activated the effector protein inside the cell. So that is a difference between the two. Now tyrosine kinase associated receptor, the classic example is growth hormone receptor. So how this receptor works is that the receptor itself may not have tyrosine kinase activity, but its cytoplasmic tail is linked to the tyrosine kinase. So instead of calling it tyrosine kinase receptor, we call it tyrosine kinase associated receptor. But the mechanism is almost the same. That is, when the hormone comes and combines with the receptor, the tyrosine kinase is straight away going to be activated. Just a slight difference between the insulin receptor and this one. Cytoplasmic tail is linked to the tyrosine kinase enzyme, whereas receptor itself having the tyrosine kinase activity, slight different. And when growth hormone binds to the receptor and then tyrosine kinase is activated, then another machinery inside the cell will be activated. It's called as JAK-STAT machinery. Janus kinase enzymes and signal transducer activated transcriptors. So that's a growth hormone receptor, and this in summary was the mechanism of hormone action for various types of hormones. Now Let me just summarize a few quick things related to the hormones, and then we will move on to discuss the individual hormones. Let's just summarize first about a few hormones and their half-life or longest, shortest hormones, etc. Shortest half-life is for catecholamines, a few seconds. Then growth hormone, short half-life, 1 to 2 minutes. Then insulin, 5 to 8 minutes, and so on. So shortest half-life. Longest half-lives, well, when I say shortest or longest, it's only one, but I am giving the hormones in that category as well. So shortest half-life catecholamines, a few seconds, and then the peptide hormones like others which I mentioned, growth hormone, insulin. Longest half-life is said to be of vitamin D, 15 days. Obviously it's a steroid hormone, so long half-life. Then next in that category, I mean, if you remove this this one is the longest one, but apart from that, thyroxine, 7 to 8 days. So that's longest half-life. Shortest latency for the onset of action. Some hormones they start acting very rapidly, very short latency. Example in this is oxytocin, a few seconds. Milk ejection reflex, it starts, see, by latency I mean from the stimulus to the onset of action of that hormone. This gap is called as the latency, latent period. So shortest latency is for oxytocin, a few seconds, whereas longest latency is for the thyroxine. The thyroid hormone has got a latent period of 48 to 72 hours. By the way, what do you understand by latency of action for thyroxine? Well, you know, thyroid hormone, its most basic function is to increase the basal metabolic rate, and that will generate the heat also as a byproduct. So when, for instance, we enter the cold environment, there would be shivering thermogenesis. There would be shivering or thermogenic shivering, and slowly one would shift from shivering thermogenesis to non-shivering thermogenesis. What is that? Non-shivering thermogenesis, when there would be increase in the secretion of thyroid hormone, and that takes about 48 to 72 hours. So this is said to be the latency for thyroid hormone action. Shortest hormone, TRH. TRH is the shortest hormone. So the short hormones, shortest is TRH. Then we have octapeptides or nonapeptides. Oxytocin, its source is a nonapeptide, and then oxytocin itself is an octapeptide. So oxytocin also is a very short hormone. Whereas longest hormone, HCG, is said to be the longest hormone. 241 amino acids, followed by growth hormone, 191 amino acids. So those are the long hormones. Well, so that is the introduction to the endocrine system.